Hello biology students, and welcome to video 2 in the organic chemistry unit. So remember in last video we talked about how we could take hydrocarbons, those carbon skeletons that have just carbon and hydrogen, and combine them into functional groups, and from that we can make 4 main types of organic molecules or macromolecules. So in this video we're going to be focusing on just the first type of molecule, which is the carbohydrate. So carbohydrates come in 3 main categories. So the first type of carbohydrate is a monosaccharide. And let's break down this name a little bit. So saccharide means sugar, and mono means 1. So a monosaccharide is going to be made up of 1 sugar. And the monosaccharide is going to be the monomer of a carbohydrate. Remember that monomer, that vocab word from last video, which meant the building block. Similarly, the second type of carbohydrate is a disaccharide, and here di means 2, so a disaccharide is going to be made up of 2 sugars, or 2 monomers. And finally, a polysaccharide, poly means many, so a polysaccharide is going to be the polymer, and this is going to be made up of 3 or more monomers, 3 or more sugars. Okay? All carbohydrates are made up of 3 main elements: carbon, hydrogen, and oxygen. Okay, so let's talk about monosaccharides first. So like I said on the previous slide, the monosaccharide is the monomer, or the building block, of all carbohydrates. Every single monosaccharide has the exact same molecular formula, and that's C6H12O6. And I'm running this again because my numbers here didn't come out as subscripts on my video. But what this means is every single monosaccharide has exactly 6 carbons, 12 hydrogens, and 6 oxygens. No matter what monosaccharide we're talking about, they all have that structure. They also all have the same general function. So these simple sugars, these monosaccharides, are mainly used as energy or fuel for the work that the cell does. And the way it powers the cell is through a process called cell respiration that we're going to see in a lot more detail later in the year. So let's look at a picture of a monosaccharide. So you are responsible for what these molecules look like. So make sure you're taking note of this. Okay. So if somehow you were able to have a monosaccharide not in a solution, what it would look like, it would look like this, and it would have 6 carbons, 12 hydrogens, and 6 oxygens. But it turns out almost all the time we find monosaccharides in some kind of solution, and when they're put into a solution, they will spontaneously change from this linear structure and they'll change into this ring structure, okay? So monosaccharides, this is supposed to be an arrow here, monosaccharides are really just one ring. So whenever you see something like this, that's a big clue to you that that's a monosaccharide. That's the main structure. Okay? There are 3 main examples of monosaccharides that I want you to be concerned with. The big one, the biggie, is glucose. Glucose is the main monosaccharide. It's the main fuel for our cells in our body. And there's also fructose and galactose. Okay? And all of these molecules have the same exact formula. They're all monosaccharides, so they're all C6H12O6, but their atoms are in different places. So for example, I want you to compare here glucose, which is this one right here, to galactose over here. So you can say, see, in glucose, I have an oxygen and a hydrogen here, and a hydrogen here, where in galactose, I have an oxygen, a hydrogen here, and a hydrogen here. So even though they have the same atoms, they're in different spots. And by putting those atoms in different places, we've given these molecules different properties. So even though these 3 molecule types have the same structure, glucose is the main source of energy. It's found in corn syrup, whereas fructose is really, really sweet. It's found in things like fruit, honey, and galactose is one of the sugars found in milk. Okay, so they all have very different properties, even though they're made up of the same atom types. So when we have a situation like this, where we have a structure that has the same number and types of atoms, so in other words, they have the same formula, but they have different structures, those atoms are in different places, we call that an isomer. So in this case, glucose, fructose, and galactose are isomers of one another because they have the same number and types of atoms, but those atoms are in different places. They have different structural formulas, and therefore they have different properties. Okay. So remember dehydration synthesis from last video. So dehydration synthesis, remember, is when I take away water, I dehydrate something to synthesize or build a bigger molecule. So if I have 2 monomers, you can see I have a glucose and a glucose, so 2 monosaccharides. if I take away a water and bond this oxygen to the carbon that's on that corner right there, I end up with two monosaccharides linked together. And what that is called is a disaccharide. Di meaning two, so this is two monomers put together. So each disaccharide is made up of two monosaccharides, and there are three main types of disaccharides that I want you to know. So sucrose, so this is the sugar that you would have in your pantry at home. It's just typical table sugar, and it's made when you use dehydration synthesis to combine a fructose monosaccharide with a glucose monosaccharide. So lactose is the main sugar in milk, and this is made up of the two monosaccharides, galactose and glucose. And finally, maltose, this is the sugar that's found in beer. It's the sugar that's mainly used in brewing, and this is it formed if you combine two glucose monosaccharides. Okay, and just like every monosaccharide was C6H12O6, every disaccharide is C12H22O11. And in class tomorrow, we'll talk about more why it has that formula, because that is actually kind of interesting. Okay, so lactose. So this is a picture of lactose. So you can see it's a disaccharide, so it has two main rings. So whenever you see two rings linked together, you know it's a disaccharide. So it turns out that lactose in particular has a funny bond. A lot of disaccharides, if you had looked back on a previous page, have a bond that looks like this. Okay, kind of like a V. Or you can see this lactose, this disaccharide, has a bond that looks like this. And it turns out that some people do not have the enzyme that can break this special bond, this weirdly shaped bond. And if you don't have that enzyme to break this down, you can't break down lactose, and that's called being lactose intolerant. So if there's anyone listening to this video right now that's lactose intolerant, it's because you can't break this simple alpha-galactosidase linkage that's right there. Okay. So then our third category of carbohydrates are polysaccharides. Remember, poly means many, so there's going to be many monosaccharides linked together. Okay, and they would all be linked together by dehydration synthesis. That's what that DS stands for. So polysaccharides have two main functions. Some polysaccharides are energy storage molecules, and more specifically, starch is a polysaccharide that's an energy storage molecule, and glycogen is an example of an energy storage polysaccharide. And then so they can store energy, or they can also be structural. They can provide structure to cells. And the two polysaccharides that you have to know that do this are cellulose and chitin. So let's look at these in a little bit more detail. First, let's look at the short-term energy storage one. So the first two ones on the list on the previous slide. So starch, this is the polysaccharide that we find in plants. So you can kind of think stuff like potatoes. You say that potatoes are a very starchy food. And that's because they're mainly composed of starch. And all the starch in that potato is all the energy that the potato plant was storing for later use. And if the potato plant wanted to use that energy, all it would have to do is hydrolyze. Remember, that's hydrolysis, the process to break up a polymer into a monomer. If it hydrolyzed, if it broke up that starch into little glucose molecules, it could then use glucose for energy. Because remember, we said that glucose is the main energy source in the cell. A similar molecule in animals is glycogen. So animals don't have starch, they have glycogen. And this is stored in your liver and in your muscle. And very similarly, let me draw. Okay, so here, it's my polysaccharide. It's made up of many monomers. So say if I'm an animal and I needed some energy, I could use hydrolysis. I could break these bonds, and then I could use these individual glucose molecules for energy, because we know glucose is the main energy source for the body. The other type of polysaccharide, remember, is a structural polysaccharide. And again, there's two main examples I want you to know. So in plants are mainly use cellulose as their structural polysaccharide. You might have also heard this as fiber. So humans cannot digest cellulose, so you can kind of think like things like celery. That's really hard to chew and break down because, I mean, we don't really have the enzymes to break down that structure, but it gives the plants a hard, firm structure so the plant doesn't fall over. Another structure of polysaccharide is chitin, and this is found in both in animals, so it makes up the exoskeleton, or kind of like the outside shell that insects and crustaceans, things like crabs, have. And it's also used in fungi as well to give them firm cell walls. Here's some pictures. So you can see, here's a picture of starch, glycogen, and cellulose. So these are all polysaccharides, and you can see they're made up of many monosaccharides linked together. And they can be hundreds or thousands of monosaccharides long. Okay, so here's a different picture, again, showing a similar thing. So this is cellulose this time. And you can see, again, that it's many monosaccharides linked together to form a long chain. Okay, so finally, the last thing we have to talk about in this video is how we regulate carbohydrate levels in our body. Okay, so we know that monosaccharides are the main source of energy. So you might wonder, well, how are we going to get monosaccharides into our body? And there's two main ways. You could eat them, so you could literally eat a pile of glucose or a pile of fructose, right? And that would put some monosaccharides into your body for you to use as energy. Or, remember, if we have some kind of storage polysaccharides, maybe if we had glycogen in our liver, we could break down that glycogen, that long polysaccharide, into individual monomers and use that as energy. Or if we, ate a potato, we ate some starch, we could break down that polysaccharide into monomers and use that as energy as well. Okay, so finally, so say you eat a bunch of glucose. You eat a big meal and you have lots of monosaccharides in your bloodstream. So you have a huge and increased amount of glucose in your blood. Well, obviously we want to have lots of sugar, but we want that sugar to not be in our blood. We want it to be in our cells so cells could use it for energy. Okay, and it's unhealthy to have lots of extra sugar floating around in your blood. So if you have too much glucose in your blood, what happens is that your pancreas, which is an organ in your abdomen, uses a hormone called insulin. So insulin, this hormone, this molecule, will leave the pancreas and it will signal the liver. So it will say, hey liver, there's too much glucose in the blood and I want you to store it. So the glucose, all that extra glucose floating around in the blood, will be linked together via dehydration synthesis. So I'll form a bond by taking away water, and I'll build a big, long polysaccharide, that glycogen, that storage polysaccharide in humans. Okay, it will be stored in the liver, and then if the liver gets full, it will be stored in muscles, and finally, if the muscles get full, it will be stored in fat. So now I've taken the extra glucose out of the bloodstream, and I've put it in a polysaccharide so I could store it. But then say maybe a few hours later, now you have a very low blood glucose because you haven't eaten a lot, your cells have used all the extra up, so you really need some sugar or some monosaccharides to, boost and give you some energy. So now what happens is the pancreas, instead of sending insulin, it sends a different hormone called glucagon to the liver, and that glucagon tells the liver, hey liver, there's not enough glucose in the body, so I want you to take that glycogen. Remember, glycogen was the storage polysaccharide, that we just made, and I want you to break it up, I want you to hydrolyze it into glucose and put it back in the blood so the cells can bring it up and use it for energy. Okay? I've got a couple of pictures of this. So if I have too much glucose, we're going to use the pancreas is going to send insulin to the liver to tell it to turn glucose into glycogen. Glycogen's the storage polysaccharide. But then if our glucose gets too low, the pancreas will send glucagon, a hormone, to tell the liver. We'll turn that storage polysaccharide back into glucose so we can bring our glucose levels back to normal. And this is an example of homeostasis. We're keeping glucose levels in the blood steady. So one more picture. This one's a little bit more complicated, but it's showing you the exact same thing as the previous slide. So if you eat something and your blood sugar is too high, the pancreas was going to release insulin, and that will convert glucose into glycogen. Okay? And then if the blood sugar gets too low, the pancreas is going to release glucagon to put that glycogen back into glucose to bring our blood sugar levels back up. So that's it for video 2, and I will see you in video 3.